Hello everyone. Today we will discuss about the metabolism of galactose. This galactose is derived from the hydrolysis of the disaccharide lactose in the intestine by the enzyme lactase. This galactose is actively transported to portal blood. Galactose absorption is faster than that of glucose. This galactose reaches liver by portal blood where it is readily converted to glucose. This property is used as a test of liver function called as galactose tolerance test. This galactose reaches liver by portal blood where it is readily converted to glucose. This property is used as a test of this liver function called as galactose tolerance test. Most of the dietary galactose is converted to glucose and goes to systemic circulation as glucose. Very little galactose as such goes to systemic circulation. So, most of the dietary galactose is converted to glucose and it goes to systemic circulation as glucose. Very little galactose as such goes to systemic circulation. So, coming to the metabolic pathway, step 1 is galactose is phosphorylated with the enzyme Galactokinase using ATP and it forms galactose 1-phosphate. Galactose by the enzyme Galactokinase here ATP, ADP, Mg++. Galactose is phosphorylated to galactose 1-phosphate. Galactose 1-phosphate. This is the first step. This reaction is irreversible. In the step 2, this galactose 1-phosphate reacts with UDP glucose and it forms UDP galactose and glucose 1-phosphate. This reaction is catalyzed by the enzyme galactose 1-phosphate uridyl transferase. So, by the enzyme galactose 1-phosphate uridyl transferase, galactose 1-phosphate reacts with UDP glucose. This is UDP glucose and it forms UDP galactose and glucose 1-phosphate. Glucose 1-phosphate and UDP galactose. Here galactose 1-phosphate uridyl transferase is the enzyme. Galactose 1-phosphate uridyl transferase. Galactose 1-phosphate reacts with UDP glucose to form UDP galactose and glucose 1-phosphate. This reaction is catalyzed by the enzyme galactose 1-phosphate uridyl transferase. This is the second step. Then in the third step, this UDP galactose and UDP galactose is converted to UDP glucose by the enzyme Epimerase. UDP galactose, it is converted to UDP glucose. This reaction is reversible. This is catalyzed by the enzyme uridine diphosphate galactose epimerase. Uridine diphosphate galactose epimerase. This is the enzyme. Here in the next, this is the third step, that is UDP galactose is converted to UDP glucose. This UDP galactose and UDP glucose, these are interconvertible and this reaction is catalyzed by the enzyme epimerase. This epimerase requires NAD+. This reaction is reversible. In this manner, glucose can be converted to galactose or galactose can be converted to glucose so that preformed galactose is not essential in the diet. So in this manner, glucose can be converted because this is irreversible reaction. So UDP galactose can be converted to UDP glucose. UDP glucose can be converted to UDP galactose. So in this manner, glucose can be converted to galactose so that preformed galactose is not essential in the diet. Then there will be biosynthesis of lactose. So this next step is the biosynthesis of lactose. In the synthesis of lactose in lactating mammary gland, UDP glucose, this UDP glucose is converted to UDP galactose. First, the UDP glucose is converted to UDP galactose by epimerase. Then this UDP galactose then condenses with one molecule of glucose to form lactose. This UDP galactose first condenses with one molecule of glucose and it forms lactose. So this UDP is released. UDP is released. So in the biosynthesis of lactose, the UDP galactose, this UDP glucose is first converted to UDP galactose by the enzyme epimerase. This UDP galactose then condenses with the one molecule of glucose and it forms lactose. This reaction is catalyzed by the enzyme lactose synthetase. Enzyme is called lactose synthetase. This enzyme is otherwise known as galactosyl transferase. Galactosyl transferase. So, formation of lactose by the enzyme lactose synthetase, otherwise known as galactosyl transferase. This formed UDP glucose then can be converted to glycogen. Can be converted to glycogen. Then by phosphorylation, by the enzyme phosphorylase, it is converted to glucose 1-phosphate. It can be converted to glucose 1-phosphate. Then this glucose 1-phosphate then reacts with the one molecule of UTP. Glucose 1-phosphate reacts with one molecule of UTP. Then here this inorganic pyrophosphate is released by the enzyme UDP pyrophosphorylase. It forms UDP glucose. UDP pyrophosphorylase. So this glucose can be converted to glycogen and by the enzyme phosphorylase, this glycogen can be converted to glucose 1-phosphate. This glucose 1-phosphate again condenses with one molecule of UTP by the enzyme UDP pyrophosphorylase. Here inorganic pyrophosphate is released and this glucose 1-phosphate it is converted to UDP glucose. Then the UDP glucose undergo epimerization and it forms UDP galactose and this cycle continues. And it forms lactose. Then coming to the glucose 1-phosphate, this glucose 1-phosphate can be converted to glucose 6-phosphate. Glucose 6-phosphate. Glucose 1-phosphate can be converted to glucose 6-phosphate by the enzyme phosphoglucomutase. Then this glucose 6-phosphate can be converted to glucose. That is a reversible reaction. Glucose 6-phosphate can be converted to glucose by the enzyme glucose 6-phosphatase. Glucose 6-phosphatase. So that this phosphate group is removed. Addition of one molecule of water. Then glucose is formed. This is a reversible reaction. So this glucose can be converted to glucose 6-phosphate by the enzyme hexokinase. Here one molecule of ATP. This is the reaction. So this glycogen, UDP glucose can be converted to glycogen, glycogen to glucose 1-phosphate, glucose 1-phosphate condenses with the UTP, it forms UDP glucose. This glucose 1-phosphate by the enzyme phosphoglucomutase, it is converted into glucose 6-phosphate. this glucose 6-phosphate can be converted to glucose by the enzyme glucose 6-phosphatase. Here, one phosphate group is removed. Then glucose, this is since it is a reversible reaction, this glucose can be converted to glucose 6-phosphate by the enzyme hexokinase. So this is the metabolic pathway of this galactose. Coming to the regulation of biosynthesis of this lactose, the enzyme is lactose synthetase, or it is known as galactosyl transferase. Coming to the regulation of this biosynthesis of lactose, enzyme is lactose synthetase. This enzyme has two subunits, lactose synthetase enzyme. It has two subunits, that is catalytic subunit and modifier subunit. Catalytic subunit and modifier subunit. Catalytic and modifier subunit. This catalytic subunit is known as galactosyl transferase. Galactosyl transferase. And this modifier subunit is called as alpha lactalbumin. Alpha lactalbumin. So this lactose synthetase enzyme, it has two subunits, catalytic subunit and modifier subunit. Catalytic subunit is called as galactosyl transferase and this modifier subunit is called as this alpha lactalbumin. This galactosyl transferase, that is a catalytic subunit, does not use D-glucose as one of its substrates in tissues other than mammary gland. In other tissues, it is involved in the transfer of galactosyl moiety from UDP galactose to N-acetyl glucosamine, and it forms N-acetyl galactosamine, that is UDP galactose plus N-acetyl glucosamine, N-acetyl glucosamine, and it forms N-acetyl galactosamine, N-acetyl galactosamine. This galactosyl transferase does not use D-glucose. It does not use D-glucose as one of its substrate in tissues other than mammary gland. So in mammary gland, it uses D-glucose as the substrate. In other tissues, it is involved in the transfer of this galactosyl moiety from the UDP galactose to N-acetyl glucosamine, and it forms N-acetyl galactosamine. Then in the lactating mammary gland, binding of this alpha lactalbumin, that is the modifier subunit, to the catalytic subunit, alpha subunit, binding of this alpha lactalbumin to, or the modifier subunit to the catalytic subunit, brings about the transfer of galactosyl moiety to D-glucose, and it forms lactose. That is the formation of lactose. That is, in the lactating mammary gland, this alpha lactalbumin or the modifier subunit, it binds to catalytic subunit. It brings about the transfer of galactosyl moiety to D-glucose, and it forms lactose. Then coming to hormonal regulation in the biosynthesis of lactose. Coming to hormonal regulation, this alpha lactalbumin, that means the modifier subunit, alpha lactalbumin level in mammary tissues is under hormonal control. The hormonal changes that follow parturition increases the cellular level of modifier subunit, that is alpha lactalbumin. This alpha lactalbumin level in mammary tissues is under hormonal control. The hormonal changes that follow parturition increases the cellular level of modifier subunit, that is the alpha lactalbumin. Then this prolactin increases the rate of synthesis of both subunits. Increases the rate of synthesis of both subunits. Then comes progesterone. Progesterone, it inhibits the synthesis of alpha-lactalbumin. At parturition, levels of progesterone decreases and alpha-lactalbumin level increases or synthesis increases. This is the regulation of the synthesis of this lactose, regulation of biosynthesis of lactose. Coming to the clinical aspects, that is inherited disorders of this galactose metabolism. Inherited disorders of galactose metabolism. First one is galactosemia. Galactosemia. It is an autosomal recessive disorder. Autosomal recessive condition. In this, there is inability to convert galactose to glucose in normal manner due to the deficiency of the enzyme galactose 1-phosphate uridyl transferase. Galactose 1-phosphate uridyl transferase. Normally, this enzyme catalyzes the conversion of this galactose 1-phosphate to UDP galactose by in the reaction galactose 1-phosphate it reacts with the UDP glucose by the enzyme galactose 1-phosphate uridyl transferase. It forms UDP galactose and glucose 1-phosphate and glucose 1-phosphate. This is the reaction of this enzyme galactose 1-phosphate uridyl transferase. Here in the galactosemia, this galactose 1-phosphate uridyl transferase enzyme is deficient. This is the classical type of this galactosemia. This enzyme deficient is, this is the classical type. In this, both galactose and galactose 1-phosphate accumulates in blood and tissues like RB cells, liver, spleen, kidney, heart, lens of eye and cerebral cortex. Here, both galactose and galactose 1-phosphate accumulates in these tissues because this galactose 1-phosphate, it cannot be converted to UDP galactose. It cannot be converted to UDP galactose, so it accumulates due to the deficiency of the enzyme. Next one is galactokinase deficiency. That is a minor type. Minor type, that is the galactokinase deficiency. Usually this galactokinase catalyzes the conversion of galactose to galactose 1-phosphate. That is the first step in the metabolic pathway of galactose to galactose 1-phosphate. So in this reaction, so in this disorder, that is galactosemia, in the minor type, that is galactokinase deficiency, this galactose accumulates in blood and tissues because this galactose cannot be converted to galactose 1-phosphate. So galactose accumulates in blood and tissues. Rarely there will be epimerase deficiency. Epimerase deficiency. So clinically, infants having this disorder appear normal at birth, but later failure to thrive, becomes lethargic, may omit and will have this jaundice, and there will be hypoglycemia. Reason for this hypoglycemia is that due to the enzyme deficiency, galactose cannot be converted to glucose. So galactose cannot be converted to glucose due to enzyme deficiency. So consequently, glucose level decreases, it leads to hypoglycemia. Then increase the galactose level because this galactose cannot be converted to glucose. So galactose accumulates. So increase the galactose level, it increases insulin secretion. Increase the galactose level, increases insulin secretion. This also lowers glucose. Then galactose 1-phosphate inhibits phosphoglucomutase enzyme. It inhibits phosphoglucomutase enzyme. Phosphoglucomutase enzyme means it converts glucose 1-phosphate to glucose 6-phosphate. Glucose 6-phosphate. So this reaction cannot take place because phosphoglucomutase is absent. So from this glucose 6-phosphate, glucose is formed. So this reaction cannot occur. So consequently, glucose level decreases. That is in case of infants. Then after 2 to 3 months, liver may show fatty infiltration and it leads to liver cirrhosis. Then mental retardation due to this accumulation of galactose and galactose 1-phosphate in cerebral cortex. Then next one is development of cataract. Development of cataract. So let's see the causes of this cataract. Excess of galactose in lens is reduced to galactose. So galactose cannot be converted to galactose 1-phosphate and will not take place the pathway. So galactose accumulates. So this excess galactose, it forms galactose or dulcitol. This excess of galactose in lens is reduced to galactose or dulcitol by the enzyme aldose reductase. This takes place in lens of the eye. Galactose is converted to or reduced to galactose or dulcitol by the enzyme aldose reductase. This galactose, it cannot escape from the lens cells. Osmotic effect of the sugar alcohol, that is the this is the sugar alcohol, that is galactose. So osmotic effect of the sugar alcohol contributes to injury of lens proteins and it causes development of cataract. This galactose, it cannot pass through the lens cells. So osmotic effect of the galactose contributes to injury of the lens proteins and it develops cataract. This excess of galactose inhibits glucose 6-phosphate dehydrogenase. It inhibits glucose 6-phosphate dehydrogenase. It leads to less NADPH, which results to less reduced glutathione, that is GSH, reduced glutathione. So it is GSH. That is the development of cataract, causes of development of cataract. Then biochemical findings include increase the galactose level, increase the galactose, consequently decrease the glucose, increase the galactose, decrease the glucose, then decrease the inorganic phosphate due to utilization of phosphate for galactose 1-phosphate. Then urinary findings include increase the excretion of galactose in urine, that is galactosuria, increase excretion of galactose in urine, that is galactosuria, then albuminuria and amino aciduria. The amino acids usually excreted are glycine, alanine and serine. This is not fatal, then can survive to puberty and adulthood. Then coming to the reasons, this epimerase reaction is freely reversible. Epimerase reaction means this UDP galactose and the conversion of UDP galactose and UDP glucose. So this reaction is reversible. Hence, even with lactose-free diets, galactosemic children can meet the body requirement of galactose from glucose. Galactose or D-galactose to D-glucose. This is catalyzed by the enzyme Epimerase. UDP galactose to UDP glucose by the enzyme Epimerase. So, Epimerase reaction is freely reversible. Hence, even with the galactose-free diet, galactosemic children can meet the body requirement of galactose from glucose. When puberty is reached, alternate pathway for this galactose may develop. Alternate pathway for this galactose, that is galactose. It is first converted to galactonolactone. Galactonolactone. Then it forms galactonic acid. Galactonic acid to beta keto galactonic acid. Beta keto galactonic acid. Then it forms D-cellulose. cellulose. From D-cellulose it forms D-cellulose 5-phosphate. D-cellulose 5-phosphate. Then this enters into HMB shunt pathway. So when puberty is reached, alternate pathway for this galactose may develop. Here galactose can be converted to D-cellulose 5-phosphate and this can join HMB pathway. So this is the related to inherited disorders, that is galactosemia. Then this is today's topic. Thank you for watching.